Hello, everyone. Thank you so much for joining us today for this TwinStrand webinar. My name is Jenna Gallegos with Samba Scientific, and I'll be the moderator today. In this webinar, Danielle LeBlanc and Francesco Marchetti from the Health Canada Environmental Health Science Research Bureau will present their work using duplex sequencing for robust detection and characterization of mutations from prototypical mutagenic exposure in human and mouse models. The talk will begin with a brief technology intro by TwinStrand Vice President of Informatics, Clint Valentine. During the presentation, if you have any questions, you can submit them using the Q&A button. Please put your questions here and not in the chat window, as this will ensure that we can record your questions and answer them either during the Q&A or with a follow-up after the webinar. Also, we are recording this webinar, so if you miss any part of it or want to rewatch or share it with colleagues, you'll be notified in a few days by email with a link where you can access the recorded video. So without further delay, I'll turn the time over to Clint Valentine.
Thank you. Hello, my name is Clint Valentine, and I lead bioinformatics and software development at TwinStrand BioSciences in Seattle. Today, I'd like to introduce a high accuracy and high throughput technology that can be used for in vitro and in vivo mutation detection studies, duplex sequencing. Classically, artifacts and high error rates associated with DNA sequencing have prevented the easy detection of very rare mutations. Common artifacts include base misincorporations during library preparation, jackpot errors amplified by PCR, and sequencer miscalls. Distinguishing rare mutations from artifacts is important, as they can be used as a biomarker for many genetic tox applications like drug and chemical safety assessment or cancer risk studies. Duplex sequencing eliminates technical noise by using a clever approach of tagging source molecules so that each sequence strand copy can later be informatically grouped and error-corrected together. You can see how this works in this schematic, in which the occasional or recurrent artifact in red is resolved through a consensus-based approach. The process begins with a biochemical step that adds duplex-seq tags to fragments of DNA from your experiment. These tags contain barcodes that uniquely identify not only the piece of duplex DNA the tag is attached to, but also the individual strands of that duplex such that they can be distinguished from one another. After DNA amplification and sequencing, duplex-seq tags let us compare copies of the Watson and Crick strands from each duplex so that we can quickly identify where mistakes were introduced, and we can reconcile base calls that don't match between the strand consensuses. The result is an average error rate below 1 in 10 million and an often perfect reconstruction of the original source DNA molecule. The outcome of using duplex sequencing as compared to an industry standard sequencing protocol is shown here. On the left, we have DNA sequencing without duplex sequencing, and the result is a carpet of genetic variation below 1% abundance, which is actually the result of technical artifacts and noise. On the right, we highlight that same territory in the genome, but using a duplex sequencing protocol, and we reveal a single true mutation and no false positive signal. So what can you learn from data generated in a duplex sequencing study? Well, for starters, the technology enables the direct quantification of DNA mutations instead of proxies for mutation. This means that you can analyze loci from any tissue, species, or organism provided they contain DNA. With mutations fully characterized, your data becomes more valuable because it helps you understand the influence of repair processes, inferred mutagenic modes of action, and the impact of mutations on cellular function, among many other interesting phenomena. But because rare mutation detection requires large data sets in which we've sequenced millions or billions of DNA fragments, a scalable bioinformatics pipeline is required. Shown here is an example of what it takes to analyze sequencing data generated using a duplex sequencing protocol. Of note is that many of these steps are standard and best practice in the field of next-generation sequencing, yet a few of them are unique and critical to duplex sequencing, such as the consensus calling process itself. The outputs of this bioinformatics pipeline are many diagnostic metrics to assess the performance of your experiment, as well as a final variant call set, which is a simple database of the mutations found in your sample. A bioinformatics pipeline like shown here is something that TwinStrand provides automated and prepackaged in a secure and compliant cloud platform so that you can be up and running in minutes after you've uploaded your sequencing data. At TwinStrand, we provide kit-based mutagenesis assays for human, mouse, and rat species, and we also have the capability for collaborative-based mutagenesis scientific projects. Products and services come bundled with access to secure cloud-based bioinformatics software, and TwinStrand has a support team at the ready in the event you need training, assistance, or help interpreting your results. At TwinStrand, we're motivated by high ideals of scientific rigor and stewardship, and we're excited to talk to you about how you might apply this technology for the next generation of genetic toxicology breakthroughs. Thanks for your attention, and especially thank you to our speakers, Dr. Francesco Marchetti and Danielle LeBlanc. I'll now turn it over to Francesco.
Thank you, Clint, for that introduction of duplex sequencing. And 1 second. And I'm very happy to have this opportunity to share what we have done at Health Canada over the last 3 years to advance genotoxicity testing by using duplex sequencing. I will go over the results that we have generated, and then Danielle LeBlanc will tell you about her hands-on experience with duplex sequencing in the lab. Before I start, I want to acknowledge the many collaborators who have contributed to the data I'm going to present to you today, and in particular, the 5 people who actually have done the work in the lab, Danielle, Gu, Annette, Unara, Elizabeth, and also Carol Yark, my longtime collaborator. So we know that genotoxicity testing is critical for health protection, and that exposure to genotoxic agents can result in important health effects that can manifest themselves long time after the exposure. And as indicated in this graph taken from Jesse, cancer is one of the major endpoint of concerns for genotoxicity testing. And this is a process that takes place over many years, but genotoxicity is generally focused on these very early phases of the process, in which we want to identify whether a chemical has the potential to result in cancer by causing genetic alterations. And then it's quickly summarized below. We are generally looking for whether a chemical can induce DNA damage, mutations, which is the main focus of this presentation, or chromosomal abnormalities. And over the course of the last 40, 50 years, the genotoxic community has generated many assays that have been used to study whether a chemical is genotoxic. And summarized on this slide are the OECD test guidelines that are currently available. And you can see that we can do tests in bacteria, in vitro, and in vivo. And on the right side of the slide, there are the 4 mutagenicity assays with OECD test guideline. And of course, they have worked well, and they're still working well, but nothing is perfect. And we are currently facing some challenges and needs in mutagenicity testing. And specifically, the current assays that are available, they have limitations, including the fact that you need to use specific rodents or cell lines to test for mutagenicity, so they cannot be easily integrated into other toxicity tests. They are limited to either bacterial reporter gene or to a single mammalian gene. They are generally assessing mutations that cause a phenotypic change. They are limited to detecting mostly point mutations, small indels. And if you want to understand the mechanism, you need to characterize the mutation spectrum, and additional work is done, has to be done to do that. So we clearly need improved mutagenicity assays that are better integrated, they're more comprehensive, they provide mechanistic information, they are human relevant and efficient, and they also contribute to the 3R effort of replace, reduce, and refine animal testing. And at Health Canada, we do think that duplex sequencing can play a big part in advancing how we assess mutagenicity. And so we have started working with the panels that were developed by TwinStrand, the mouse mutagenesis panel and the human mutagenesis panel. And both panels are composed by 20 targets regions distributed across the autosomes, and they're formed by a combination of gene targets and non-gene targets. And we are using the mouse mutagenesis panel for in vivo studies and the human mutagenesis panel for in vitro studies. And the goal of these studies that we are doing is to really look at what is the best study design, also look at cross-laboratory concordance of the assays, its sensitivity, and also what kinds of mechanistic analysis. For the in vivo studies, we're also then looking at tissue specificity, and most importantly, at the concordant with the plaque-based TGR assay, which is the current gold standard for mutagenicity testing. So I will describe first the studies that we have done in vivo, and we're using the MutaMouse, which is the transgenic model that we have been using for more than 30 years at Health Canada. And what we have done are exposing mice for 28 days to either benzoylpyrene, procarbazine, or ENU, and then collected either bone marrow or seminiferous tubules from the exposed animals. We learned quite quickly that the DNA isolation that we're using for the LAGZI assay was not conducive to good DNA for duplex sequencing. And so what we're doing now is to separate the DNA, the tissue, and process half for DNA isolation with the QIAGEN kit to do duplex sequencing, and the other half we do the standard phenol chloroform to conduct the LAGZ assay. And then we applied the mouse mutagenesis panel and the bioinformatic flow, and these are the results that you get. And here I'm showing you the first results that we got with benzoylpyrene in bone marrow, and these were published in BNC Genomics in the summer of last year. And what I'm showing you in this graph are the doses that were given. Each column is a different mouse, and the number of mutations that were identified in each mouse. And you don't really need a statistical test to really see that we got a beautiful dose response with no overlap in doses. We have now conducted a second study with procarbazine, and this work by Annette, and the paper, it's basically in the final stage before submission. And note that I've indicated with the red bar the fact that the procarbazine response is pretty much all contained within the low dose of benzoylpyrene. So the response is much weaker than what we got with benzoylpyrene. Nevertheless, even in the case of procarbazine, we got a very nice dose response. I have to point out that what I'm showing here are only unique mutations. So in those cases where we've observed the same mutation happening multiple times within the same animal, we assume that to be derived from a clonal expansion of a single mutation, and we counted here as a one mutation event. We then wanted to look at cross-laboratory reproducibility, and this graph shows the result of the benzoylpyrene samples in which DNA isolation was conducted at Health Canada, while library preparation sequencing was done at TwinStrand, versus when both DNA isolation and duplex sequencing library preparation were prepared at Health Canada, and sequencing was done by Somagen. And you can see that in those cases, there was a very good correlation between the two sets of mutant frequency, showing that duplex sequencing really has a good cross-laboratory reproducibility. Of course, what we were most interested was to compare duplex sequencing with the results of the LagZ assay that we conducted in the mutant mouse. And at the bottom, I'm showing you the results of the benchmark dose modeling, where you can assess how strong the response of a chemical is. And if you can see, you can note first that the confidence interval by which BND is analyzed is much wider for the LagZ assay with respect to duplex sequencing. So with duplex sequencing, you get a reduced intragroup viability. But when we're using only a unique mutation, you can see that the two intervals did not overlap, which suggested that the LagZ assay was more sensitive than duplex sequencing. However, when we considered all mutations, including those derived by clonal expansion for both benzoylpyrene and procarbazine, we got a stronger response. And now the confidence interval of the two BND values overlap between the two assays, suggesting that you can't really say that one assay is more sensitive over the other one. One of the major advantages of duplex sequencing is that you're not focusing on just one specific gene, but now we're looking at 20 targets spread across the genome. And what I'm showing you here are how each target responds, and a range going from the target that showed the highest response to the target that showed the weakest response. And you can see that in the case of benzoylpyrene, there was a 5-fold difference in the induction of mutations between target on chromosome 14 and chromosome 3, and there was a 2.5-fold difference for procarbazine. The other thing that you notice is that the top 6 responder targets are all intergenic. And what we found was, for both cases, intergenic mutation frequencies were higher than genic mutation frequencies. And we hypothesize that this is due to the protective effect of transcription coupled repaired on the transcribed regions. The other thing that we were very interested in observing was the fact that both the top and the bottom responding targets there was significant overlap between the two chemicals, which work by completely different mechanisms of action. And I will come back to this point in another slide. The other advantage of duplex sequencing is that while generating mutation frequencies, you're also obtaining at the same time information on the types of mutations that are induced. And as shown here, in each graph, we're going from controls to the IDOS, going from left to right. You can clearly see that the pattern of mutations induced by the two chemicals is completely different. And in the case of benzoylpyrene, you have induction of mostly C2A mutations, which are the blue mutations over here, while with procarbazine, you have induction of T2C mutations and also C2T mutations. You can then take this analysis an extra step, and now instead of just considering the mutated base, you can also look at the flanking bases and do a cosmic-like type of analysis. And even when you do that, you can see that the pattern of the signature of benzopyrene is significantly different than the signature from procarbazine. And when you start comparing that pattern with the non-cosmic signatures, what you find is with benzopyrene, you pick up signatures that have been associated with tobacco smoke, and benzopyrene is a main component of tobacco smoke, while for procarbazine, you have mostly signatures associated with defects either in DNA polymerases or mismatch repair. And as Clint mentioned in his presentation, this kind of information can give you an idea of the mechanism by which the chemical can induce mutations. A main interest of my lab is germ cells, and we also have done therefore studies with duplex sequencing looking at germ cells. And in the first study done by Danielle and Gu, we had done a dose response study with ecNGS and collected cells from the seminiferous tubules. And this slide shows the results that we've got with duplex sequencing versus the results with the LagZ assay. And in both cases, you can see that the response is very similar and that we got a significant effect with both assays. And so we were happy to see that, because one thing that you may not notice, but the response, the baseline mutation frequencies in germ cell, it's lower than what we have in the bone marrow. And so this one shows that even at this level, we are talking 10 to the minus 8, 10 to the minus 7 mutations, one mutations per 10 millions or 100 millions. Duplex sequencing has the sensitivity to pick those very rare mutations at those very low frequencies. As in the case for the bone marrow, we also saw significant differences in the response of the targets. For ecNGS, it is even more dramatical, the preponderance of mutations in the intergenic regions. And now we have these 3 targets on chromos 8, 14, and 17 that are among the top 5 responders for all chemicals that we've tested and for both tissues that were tested. So there's something intrinsically susceptible to these targets, to mutations. And we're trying to understand what may be those features that make those targets particularly sensitive. Another very interesting thing that came out from the analysis in the germ cells is that target 2 in seminiferous tubules has a significantly higher spontaneous mutation frequencies than all other targets. And this is a finding that now we have replicated in multiple groups of animals. And Daniela spent some time trying to understand, if we could see by looking at the sequence, what is making this target so sensitive and why it's only in germ cells. And of course, we don't have an answer right now, but that's something that we're really interested in understanding. Not everything works perfectly with duplex sequencing. Of course, there is a learning curve, and we got one surprise, which is when we tested benzopyrene in seminiferous tubules. When we conducted the first pilot, we had a very nice response with LagZ, but absolutely no response with duplex sequencing. So after discussing a little bit what may have been going on with TwinStrand, we decided to try to increase the DNA input that we used and double that, going from 500 nanograms to 100,000 nanograms. And when we did that with 3 different mice, we now got a response with benzopyrene as well, which indicates that for germ cells, when the level of mutation induced is very low, you may need to start with a higher amount to really capture all the mutations that are present in your samples. So to summarize the results with duplex sequencing in vivo, duplex sequencing performs well versus the gold standard in bone marrow. It has high cross-laboratory concordance, and it has the sensitivity to detect rare event, and also it appears to have low sample-to-sample viability, at least much lower than the LAG-Z assay. The ability to look at mutations across 20 endogenous loci, it gave us the ability to see how different regions respond, to demonstrate that there is really, the genome is not the same, different regions respond differently, and that non-transcribed loci have higher induced mutant frequency, and that the LAG-Z is the most responsive loci. And in future study, we're actually going to sequence the LAG-Z with duplex sequencing to have a more direct comparison of the response of the LAG-Z or LAG-Z with endogenous regions. By using this panel, you can get insight into genomic features that affect mutation induction, and you can also start to elucidate the mechanisms that may underlie cancer development. And as I mentioned before, detection of small increases, especially in germ cells, may require a higher amount of DNA than somatic tissues. I'm now going to go quickly over the results that we have obtained with in vitro using the human mutagenesis panel, and these are analyses conducted in TK6 cells. And it's an experiment that actually came out of a conversation that took place at the ecNGS. We and Innovate conducted an experiment independently of each other, and then we realized that we have done pretty much the same experimental design, and that really gave us the opportunity to do a very detailed cross-laboratory validation. And so this is what we've done, and the data that I'm showing you is presented in a paper that is currently under review as scientific reports. And quickly, what we've done, we have exposed the TK6 cells to ENU, and then collected them multiple times after the end of exposure. And then we conducted duplex sequencing on these samples. In the next few slides, you're going to see a consistent, remarkable consistency in the titles, because that's what we have found, that the results are incredibly consistent between the two laboratories. And in this slide, we start with the mutation frequencies and how they behaved across time and between laboratories. And you can see that actually, a little bit surprisingly, we did not see an impact of the sampling time, and the 48 hours seems to be already sufficient to detect an increase in mutations with ENU. And when we looked at the correlation and mutation frequencies between the two laboratories, they were highly concordant. Also, how the different loci behaved was remarkably consistent between the two laboratories, and in here the targets are shown from the least responsive to the high responsive. And as it was in the case in vivo study, we also saw in this case 30% higher mutation frequencies at intergenic loci, and also, as I mentioned, similar ranking of the targets both spontaneously and after exposure to ENU in the number of mutations that we detected. Also very consistent was the types of mutations that were detected by the two laboratories, and that didn't matter whether we were looking at mutation frequencies or at the proportional mutations. The images were absolutely identical. We did note that the pattern of mutations that we found in vitro was enriched mostly in situ substitution, which is unusual for ENU, and it is a variance with the mostly T2C and T2A substitutions that are found in vivo. And then we realized that this is due by the fact that TK6 cells are lacking the AGT enzyme that it is necessary for repairing O6 alkyl-guanine residues, which are one of the main adducts created by INU. And that shows that you really need to understand the cell type you're working with to really get a complete picture of the mutagenesis that you are obtaining. I just wanted to quickly show one result that, as the title says, fresh out of the oven. And this is work of another master student, Elizabeth Huliganga, who repeated the same study with TK6L, but now exposed to 4-NQO. And she observed a very nice dose response increase in mutation, which reached a threefold increase at the high dose. And also, when she looked at the partner mutation, we saw an increase in C2A transversion. So we now have a second chemical for which we can show that analysis of mutations 48 hours after exposure is sufficient to detect an increase in mutation. Summarizing the results that we got in vitro with duplex sequencing, the results are showing that duplex sequencing can precisely quantify in vitro mutation frequencies, that the results are highly concordant across laboratories, that 48 hours post-exposure may be an optimal sampling time for in vitro mutation analysis in TK6 cells. It works for BAP and 4-NQO. We did not get an effect with potassium bromate, which is a difficult chemical to detect with genotoxicity assay. And compared to the available gene mutation assays with the OECD test guideline, duplex sequencing, it's really provide multiple advantages, both in terms of increased efficiency, diversity of information, but also how quickly after the exposure you can get information. So where are we going next? We are conducting additional validation studies with multiple tissues, multiple chemicals. As I mentioned, we are going to sequence both the LAGZ and the PIG genes with duplex sequencing to really allow us to have a much direct comparison of duplex sequencing with the respective OECD test guidelines. We're also looking at technical studies to look at in details, for example, how many leukocytes do you need to analyze for generated data to use for regulatory applications? How many reads? How much inputs do you need? And all of these is tailored toward helping with regulatory application and adoption of duplex sequencing. And obviously, we are not doing these in isolation. We are part of a large group of investigators who are members of the genetic toxicology technical committee, who are all working together to really develop duplex sequencing and other error-correct and next generation sequencing technologies as alternatives to existing test guidelines. And we have developed this very schematic roadmap that will hope to guide the work and lead to acceptance of these guidelines. And a commentary on this topic was just published yesterday on Nature Reviews Drug Discovery. And this is the OI if you are interested in reading more on what we and others within the GTTC are doing. And with that, I thank you for your attention, and I'll stop sharing and pass the baton to Danielle.
Thanks, Francesco. Hopefully everyone can see my slides and hear me. So I'm really excited to be here today to kind of give you the insider perspective on duplex sequencing. So my focus for the past little while has been on implementing and evaluating duplex sequencing as a mutagenesis tool. And I'm going to kind of give the wet lab and data manipulation and output perspective here. Let's see. So first, I thought I would start with kind of my background from a wet lab perspective. So I do have experience with mutagenicity testing, so specifically the LACSET assay or TGR assay, and quite a bit of DNA isolation experience. Now, the LACSET assay is a lot of single channel pipetting, plating, and manual counting. It's very different than what a library preparation protocol would be. So I, previous to duplex sequencing, had no experience with library preparations and no experience with beadwork. So oftentimes magnetic beads are used in library preparation protocol to bind the DNA to allow for washing steps or isolation steps. And these can be quite finicky. So this is actually something I was quite worried about starting to work with. Luckily, TwinStrand has worked with multiple labs, Health Canada included, to kind of beta test these library building kits to make sure that not only do they work in TwinStrand's hands, but they also work in other laboratories as well. And we learned a lot during this beta testing process. So we felt like we were with TwinStrand from the very beginning. We learned a lot from them, and hopefully they learned a lot from us too. So a few of the things we learned, one was an in-person specialist is key. So very early on in the process, TwinStrand was able to send us a field application specialist that could come into our lab, see what we're working with, see the layout, see the equipment we have, and see what things might be different based on TwinStrand's lab and where they have the laboratory kits working, and see what's different in our lab. So we thought this was amazing. I don't know how many people have dealt with customer service over the phone when you're trying to get a protocol working or working with equipment that's not working. But nothing really beats a person that's there one-on-one with you to help work out any issues. So that was really great. And during the process as well, we learned of a lot of QC checks that you can use along the way during this protocol. So it's a 3-day protocol typically. And you have a lot of points where you can check that you have DNA coming out of a reaction or a step and DNA going into the next step. And this is just really peace of mind that after 3 days of being in the lab, that you're going to have something at the end and you're not kind of just crossing your fingers that it's going to work out. So those QC checks, I highly recommend when you're starting this protocol, and even doing them after you're familiar with it as well. Something we also learned, so we were doing this beta test in Ottawa during the winter. We already know it's cold and dry. This is what we look like when winter strikes, in Ottawa at least. And we never really thought of how humidity and the dryness and the cold might affect the library preparations, and specifically the beads. So the beads that you're working with, there's often steps where you have to dry them. If they get over dried, they can really affect downstream processing. You can lose DNA, and it can affect overall library preparation success. So something that may have taken 5 minutes to dry in TwinStrand's lab would take, maybe 2 minutes or less in ours. So this is just an example of some of the things that need to be considered when these kits are going to other labs and what might be different. And luckily, us and other labs and TwinStrand have taken all these results from other labs and built kits that work wonderfully now. They're 100% consistent in our hands. We have new students coming in, we're training them, even with no experience with library preparations. We're getting libraries built at 100% success, meaning that every sample that goes in, we get a library that's ready to be sequenced at the end. So that's really great. And this is just an example of some of the things that have gone into making these kits so great. So I'll just kind of go through the general protocol and workflow still on the wet lab side of things. So timing and overall protocol experience. So I found beadwork really easy to grasp, even if I hadn't worked with them before, aside from the humidity issue, which has been dealt with. I actually quite enjoyed it. I think some people cringe when they hear people enjoying lab work, but there are those of us out there who really like it. There are numerous incubation steps throughout the protocol. that allow for other work to be done during the process. So this is really nice. If you have some emails that need to be checked, or you have a last minute meeting, or you have other work that needs to be done, there are a lot of breaks within the day that it can allow you to get other stuff done. They also have overnight incubation steps that can extend up to 20 hours. So I also found this gave a lot of flexibility. I don't know how many of you have worked with other protocols where you have to stay really late in the lab and then you have to be up at 6 a.m. to check things. Some things can be really not generous in terms of time, and I found that this protocol really is. It's easy to scale up this protocol. So typically it's done in a 96 well plate with a multi-channel pipette. So when we would start this protocol, we typically start with 8 samples, especially when we're training someone new. But because of how you do the assay, it's essentially just adding more samples is adding another row that you're adding reagents to. So it's really easy to go up to 24 or even more samples at a time in a 3-day protocol. So the typical workflow is 3 days. I've just identified, you know, the general outline of how the days are, how many hours you're spending in the lab. And again, so the day 1 is your longest day. It's about 5 to 6 hours, but there are breaks in between this for other work to be done, as I mentioned. And the 2nd and 3rd day are half days, with the 3rd day essentially being a repeat of the 2nd day. And that redundancy is actually really nice because it's not 3 completely different days of new techniques that you're doing. It's very easy from a training perspective. A couple other important things to note in the protocol: there are multiple steps where you can actually pause and totally freeze the library building process by putting your plate in the freezer. And this is really good for emergencies that come up. God forbid you get COVID, you aren't allowed to come into the lab. You know, times are hard now with sickness, so it's really great to have a protocol that kind of works with your schedule and is flexible. Now data, everyone's favorite stuff. So I've covered wet labs. So you get your libraries built, you'll end up getting them sequenced. Typically you'll get FASTQ files that come out of that. And these can be put right into the Dnexus portal. So this is an online portal. It's very easy to use. TwinStrand has a Mutagenesis app on this portal that your DNA can be run through, or your FASTQ files, your data can be run through, and you can get data out of it very quickly in a familiar format. So a lot of you are probably familiar with mutation frequency data, but you also get more detailed data at the same time. And this is with no bioinformatics required. So a lot of bioinformaticians, as people know, have a lot on their plates. Sometimes they're kind of the limiting factor in terms of getting data out of what you've put in. So it's really great to have data without needing a bioinformatician to look at things. And again, the Dnexus platform is simple and easy to use. I'll just kind of briefly go over it. I'm not going to do a full tutorial just in the interest of time. So I've kind of just taken screenshots of what you're going to see when you're working with Dnexus. So I have kind of Dnexus open here. You'll log in with the username and password. You'll have all your projects laid out. And when you open them and go to output, this is similar to what you're going to see. So you'll have a folder for each individual sample. But what we're going to focus on is the mutagenesis report that you get out of it. So these are kind of the summary reports that you're getting out of your data. So if you open the mutagenesis report, you're going to see something that looks like this. So you have a nice summary of how many libraries you put in, the maximum, minimum mutation frequency, and then you have it detailed by sample as well. You have your duplex bases, mutant duplex bases, which give you your mutation frequency by sample. These can be copied, opened up in Excel, so you can manipulate them and make your own figures if you'd like. There's also a graphical representation of mutation frequency that's output as well. And I know that it's really nice when you put data into something and you get results right away that you can interpret and say, did we get a response or not? And this is, again, without anyone needing to really dive into the data and start manipulating things from a bioinformatic perspective. So this is really helpful. In addition to mutation frequency, at the same time, you also get mutation spectra information as well. So I apologize that some of the writing is a bit small, but it's not really important. So you have proportion and count of mutations types that are provided to you, data in your mutagenesis report. You're going to get detailed data that's gone into this figure for each one of your samples, how many of that specific mutation subtype you saw, what proportion, what's the background count. And again, this can be copied, it can be opened in Excel so you can manipulate it yourself as well. So this gives a bit more mechanistic insight and maybe mode of action information on the exposure you're looking at. And on top of that, within the mutagenesis report as well, you also get trinucleotide frequency information. So that's the flanking base on either side of the mutated base. And so for each of your samples, you get a nice trinucleotide context plot similar to those cosmic signature plots. And again, you can click view plot data and you get the detailed data behind those plots that you can manipulate yourself, open up in Excel, which is really useful. Now, should mutation frequency, spectra, and trinucleotide frequency not be enough, or you're looking to dig deeper into the data, you also get raw data in a usable format that can be opened up in Excel right on DNAnexus. And if you have a bioinformatician that has some time on their hands, it's very easy for them to take this data and do any number of detailed analyses that you should want, similar to the stuff that Francesco showed you. So diving into specific target differences and what genomic features may influence mutation susceptibility. Similar to what we've done in our paper, if you haven't read it yet, I highly recommend it. I might be slightly biased, but I think it's a great paper. So in summary, giving overall advantages of duplex sequencing and how personally we found it at Health Canada. So from a lab perspective, it's great that DNA can be extracted from any tissue and you can use wild type animals. So you're not reliant on specific transgenic mouse models like we have been in the past. DNA can be extracted from human cells or tissues, so you're not restricted to bacterial cells. The library kits themselves are easy to use. They've done that beta testing for you. They require no previous kit building experience. The workflow is very efficient and it's flexible with multiple schedules. From a data analysis perspective, the assay provides familiar data from what you would normally get. So you get mutation frequency, but you also get mutation subtype and trinucleotide frequency data at the same time with no bioinformatics experience required. And you also have the raw data there should you want to do any more detailed analysis. From an overall relevance perspective, we like that the mutagenesis is measured across the genome in a representative set of targets. As you've seen in Francesco's presentation, we do see quite a range of mutation frequencies across different targets. So it's really important to capture that when you're looking at mutation frequencies. And it also allows for detailed analysis into the genomic features that can influence mutation susceptibility, like transcription coupled repair, for example. And that's everything for me. Thanks so much.
Thank you, Danielle, and thank you very much to all of our speakers. We do have some questions that we're going to go ahead and answer now. As a quick reminder, if you have additional questions that you would like to ask, you can submit them using the Q&A tool at the bottom of your screen. All right, so first question from the audience. How did phenylchloroform extraction affect the duplex sequencing results?
So I can answer that. What we found was that the background mutation frequency was unreasonably high with respect to what we were expecting. We were going closer to 1 mutation per 100,000 base pairs, which was unrealistic, and at least an order of magnitude of the experience that TwinStrand had, and more than an order of magnitude of now the mutant frequency that we get now that we have shifted to the DNA isolation kit. And the idea was that the phenol chloroform extraction was too harsh and was creating additional damage into the DNA that interfered with the sequencing and resulting in a higher mutation frequency, in an inflated mutation frequency.
Thank you, Francesco. Anything from the TwinStrand team to add to that?
We've seen similar results. Phenylchloroform can be really harsh on DNA. We end up observing structural issues with the DNA molecule itself, nicks, adducts, that can be hard to overcome. So yeah, I would agree. There are better ways, and we wouldn't recommend using that solution.
All right, thank you. Next question. Has anyone started to consider whether it'll be possible in the near future to do GLP compliant environments or studies with this technology?
This is Aaron from TwinStrand. I can take that one. Yes, I am familiar with at least one CRO that is starting to work on that now. The software is already GLP ready, so it's just a matter of implementing a GLP wet lab protocol. So yeah, if you're interested, let us know. We can point you in the right direction.
Thank you, Aaron. Next question, does the assay handle just a sample of genomic regions, and how were those regions selected, and are they representative?
Yeah, these panels for all three species, they were designed to target somewhat specific places in the genomes that are scattered across the autosome, but not in a way that is ungenome representative. So we looked at a few things. We looked at nucleotide composition. We looked at the ratio of targets that were genic versus non-genic. We avoided areas that we knew were involved in negative or positive selection, so carcinogenesis processes. Ultimately, the goal of the targets that we selected and put into these panels were to find a constrained set of targets that were as genome representative as possible. And I think what we are observing with some of this data, like Francesco and Danielle have shown, is that these targets are responding differently. And that proves that no matter where you look in the genome, you might get a slightly different picture of a mutation response. So it's important to have an assay that is broadly capturing the effect of that mutagen or that exposure on the genome. And this is our best effort in doing that in a way that is constrained to just a few targets that are scalable on modern sequencing machines.
Thanks, Clint. Next question, for in vitro duplex sequencing in TK6, was cytotoxicity a limiting or interfering factor other than reduction in cell number and total DNA amount?
Yeah, of course, I didn't have the time to present everything, but we did do a dose response study to assess cytotoxicity and see which was the top dose we could use and still have a sufficient number of cells dividing. And the top dose we selected was one that it was giving roughly 50% of survival, which is what the OECD guideline asks you to do in terms of cytotoxicity. So, and of course, yes, if you have treatment that is very cytotoxic, that will affect your ability to do sequencing.
So one of the attendees pointed out that bezopyrene has increased DNA binding at CpG sites. Are there many CpG sites or similar targets amongst those that are analyzed in the assay?
Yeah, and this is a nod back to that previous question. When we design these panels, we're looking for a genome representative set of CpG sites. So we look for nucleotide composition that mirrored the genome as a whole. We also look for trinucleotide compositions that mirror the genome as a whole. So there are some targets that are going to have more CpG sites. There are some that are going to have less, but overall, they'll have a similar frequency of them as compared to the genome at large.
And just to jump in, so although they are relatively balanced in terms of GC content, We did in our paper when we looked at BAP in the bone marrow, wanted to see if elevated mutation frequencies were correlated with those targets that had the highest GC content, and we actually didn't see any correlation there, so that was a bit of a surprise.
The next question is about the sample extraction procedure. Do you recommend agarose plugs if you're concerned about introducing erroneous DNA damage? Not sure if that's a question for Danielle or the TwinStrand team.
If it's a question for me, I have never worked with agarose plugs, so I would pass that to someone that has.
Yeah, we would feel free to connect with us after the seminar, and we'd probably be able to advise based on your experiment. I think we've done experiments like this in the past, so we could probably lean on that data.
So there's another audience member that was wanting to maybe circle back on the results for the benzoate pyrene in vitro testing. Can I maybe ask you to pull the slides up with those results again? And then the question was whether or not an S9 fraction was used.
We didn't. Can you hear me?
Mm-hmm.
Yeah, we did. I didn't show the results. I just gave it as a summary, so he didn't miss it. I did not show it. And again, yeah, we can definitely share it with him if he gets in touch with me.
And we've got this information recorded too, so the questions from Alexander, we'll make sure and follow up with you on that. All right, just time for a few more questions here. We're at the top of the hour, so maybe something a little bit more high level. If this technology was widely adopted, what impact do you think that it would have on drug development pipelines, both in terms of safety and timelines? Maybe starting with Francesco.
Well, as I think this presentation showed is that you get richer data from duplex sequencing than you get from the other analyses that are currently available. So you can really start looking at, it's not just a matter, is my compound mutagenic or not? Like it's pretty much what you're getting with the current assay, but you can also start looking at, okay, is it mutagenic? What is the mechanism by which is occurring? And one thing also that we didn't present here, but it also was shown in the first paper published by Clint in PNAS, and also it's described into the commentary that I mentioned. There is another possibility of carefully selecting the targets that you're sequencing and focus, for example, on cancer driver mutations. So you can start looking at what is happening in cancer driver genes. And so you can also start getting closer to the question of whether the chemical can result in carcinogenesis. And you can get this information much earlier during the development of the drug and without conducting the very time-consuming rodent 2-year bioassay.
All right, thank you. That is all the time we have for questions today. If we did not get to your question, then the TwinStrand team will coordinate with Danielle and Francesco to be able to respond to you afterwards via email. If you have additional questions, feel free to visit the website or reach out directly at duplexseq@twinstrandbio.com. Just a reminder, this webinar was recorded. It will be available for on-demand viewing, and so you will receive an email with a link to that on-demand video in a few days. Thank you so much again to our speakers, especially Danielle and Francesco, and thank you all for attending and bringing your great questions to today's webinar. Goodbye and take care.